Okay, so I'm going to give the first of the three talks that we'll have really for the whole workshop. And my goal in doing this is to try to put together a bit of context on what it is that we're trying to do here, a little bit of history, a little bit of ideas about challenges, and the reason we're so excited about the topic and why we brought it up in the first place, which is that data and modeling have led us to think that this is an important area to focus on. Before getting rolling, I should just mention I'm at the University of Pittsburgh. I helped start a company called Immunetrics. John Bartels from Immunetrics is here, and so I'm just putting that up there as a potential conflict. I will discuss a couple things we've done with them. They've been really terrific collaborators for close to 15 years. So this is a recap of what Judy showed in her talk. So our goal is to survey and organize what's currently known about lymphoid cells and their mediators in acute inflammation, and then what has been done to date on computational modeling. And my talk is going to be trying to give a very a brief but systems-oriented overview of the inflammatory response, and also to talk about what's happened with regards to computational modeling of inflammation. It's unfortunate that two key players in the history of inflammation modeling, my good friends Gary Ahn and Yannis Androulakis, so Gary at University of Chicago and Yannis at Rutgers, were unavoidably unable to come. Gary's sick and Yannis tore a meniscus all within 2 days of this meeting. So it's really unfortunate because their insights and participation would have been really great. I'm hoping that one or both of them is listening to the live stream and will contribute. Okay, so I'm going to give a very brief background about sepsis and trauma. Tim Billier is going to talk about that background in more detail. So I'm just giving this just as a way of setting the stage. Sepsis is fairly common. If you look up the word, the septicemia in terms of cause of death, it's number 10, but in reality, sepsis happens much more than just under that designation of septicemia, so it's really more common than that. This is old, at least 750,000 cases, probably substantially more than that by now. This is from Derek Angus's work. Derek's our chair of critical care medicine at Pitt. It's mostly in the elderly and very young. And the incidence will continue to increase pretty much for the foreseeable future. Sepsis is a situation where an infection can become no longer contained in a localized fashion and can lead to acute inflammation, which has an associated with it immune dysregulation. Eventually, at some level or another, these processes interact to drive multiple organ dysfunction, a sort of cascading organ failure, and what's being called now persistent critical illness. And again, Tim's going to talk more about some of the details of what this actually entails, but think of it as a medium to long-term state that can last days, weeks, and sometimes longer, and can sometimes persist even post-discharge from the hospital. And that's something that there's a lot of interest in right now, how these early events following infection or injury can lead to long-term sequelae. So this is associated, at least in sepsis, with fairly high mortality, high morbidity, as I said, especially long-term post-discharge, and high costs. These estimates of $16 billion are really out of date by now per year, and this is only in the US. Trauma is actually fairly similar. It's the leading cause of death for people in their most productive years. Patients that survive, and we focus on blunt trauma, obviously you also have to think about penetrating trauma and more military type traumatic events that have their own set of characteristics. But in the civilian setting, blunt trauma, I think, is the major cause of this type of morbidity. Also, much like sepsis, associated with an acute inflammatory response, immune dysregulation that predisposes you together in some fashion to nosocomial infection or secondary infection that can itself become sepsis, multiple organ failure. So very many of the exact same characteristics that happen in sepsis, just often with different timing and different specific features that had previously led people to think that these are two completely different things. I'm going to try to make the case from a systems view that maybe they're actually much more similar than what people have been thinking. Just because of different circumstances, you get different dynamics and sometimes different outcomes. And so the net result of all these is what we're starting to call injury-induced persistent critical illness. So very similar type of features and phenotypes. And this long-term morbidity. So starting about 7 or 8 years ago, Gary Ahn and I, in starting to work collaboratively on trying to model the acute inflammatory response in sepsis and trauma, found ourselves in a kind of strange situation where the approaches we were trying to take, which we thought were standard computational modeling, engineering type approaches, we found that those were confusing to the two constituencies we were trying to most interact with. They were confusing to the computational people because we were thinking about clinical things, and they were confusing to the clinicians because we were thinking about computational things. And as you could imagine, that put us in this enviable position of being at the absolute lack of intersection of everyone's Venn diagram. So we tried to come up with some ideas about what made it different. What is it that we were doing that was different? And we stuck this buzzword on it of translational systems biology of inflammation, and we published this paper in PLOS Comp Bio in 2008. We were thinking about inflammation, and the first thing that we wanted to do is to try to put clear terms about what this really means, because without a clear understanding, and with trying to take as much of the bias out of this very early process, which, as I just showed you in my prior slides, is a key piece of how the body responds to infection or injury, it's difficult to make and to carry out good computational modeling work. So if I had my first take-home messages, it's objectivity. In many ways, modeling is objectivity. Because when you put the pieces together in models, if you get back the things that you see around you, that makes you believe that you put together pretty reasonable mechanisms in your models. That's something that's very hard to do in biology otherwise. Because we can always have a discussion where my idea versus your idea, whichever one of us has some data to support it, you know, who's to say who's right or who's wrong? A model can act as a kind of arbiter of which hypotheses better fit the data than others. So objectivity is a big piece of that, and we have to think objectively about what it is we're trying to actually study. So what is inflammation? It's the way the body tells itself that things are changing or have changed, either from the outside acutely, such as an infection or a trauma, or chronically, slowly, as a part of a degenerative process. It's a highly evolutionarily conserved process. It's complex and it's redundant, and it's highly interconnected and nonlinear. And it's necessary for things like healing from any sort of injury. And so when I show this, I defy anyone around to look at these features and say, wow, I mean, I really want to make a drug to stop that, right? But we do, because that's what the entire class of anti-inflammatory drugs is about. Because there's an inherent bias that says inflammation is bad. But the point is inflammation is predominantly good, which is why we have it in us, and we're set up structurally in our tissues to do inflammation locally, because it's the way we tell ourselves to fix the problem that's incipient. Now, the reason that there is a lot of money, time, and effort is spent on making anti-inflammatory drugs and therapies is because inflammation is deranged and goes bad in many disease settings. And so it's a puzzle, because inflammation could be either good or bad, or both, depending on the time of day or the particular phase of a disease, or this patient versus that patient. And so our goal is to try to go from some sort of understanding or examination of dynamic patterns of what's going on with regard to inflammation, to try to use modeling to figure out mechanism, and then to go forward from there to modulate inflammation in a rational fashion where you'd preserve the good parts of inflammation while minimizing the harmful parts. And patterns could be many things. We look at protein-level Luminex data, which I'll talk about, but we and others, big consortia such as the Trauma Glue-Grant Consortium, looked at DNA microarrays to look at transcriptomic analyses. There's the Physiome group and other people that have looked at physiologic waveforms as a function of time, and that's a huge area of interest, especially to the Department of Defense, which is the acquisition of signals from bedside monitors and using complex systems tools to and modeling tools to be able to get a lot of information about what's going on to the patient. And then the mechanism we get at with various data-driven and mechanistic modeling tools like principal component analysis, various types of analyses of dynamic networks, and then mechanistic mathematical models. So our goal is to go from a reductionist view of inflammation. So if you ask anybody about their favorite mechanism, you could probably find it in here. You know, is it all about innate immunity? Is it really all about adaptive immunity? Well, it's really more about how you actually got injured in the first place, or other people will argue it's your genetics. The point is that the answer is yes, right, to all of that. But a picture like what I just showed you is useful, but not all that useful, because it's static. It says that all I need to do is know something at a static time point about someone's genetics, and I can figure out what's going to happen next. And I think that in the long term is doomed to have limited success, because what you need to be thinking about is the process, not so much the pieces. And so we can think of this as a systems approach, and I'll show you two cartoons that have worked really well for us for well over a decade to think about how we respond to infection and to injury. So let's start thinking about infection first. So there's some bacteria, for example. They set in motion what people would call a pro-inflammatory response. Actually, we have many better detailed names for this, but I'm trying to use the most generic and easily understood terms. This pro-inflammatory response is there to be able to kill the pathogen. So already you have the beginnings of a nonlinear response, okay? This is, by definition, nonlinear. You're going forward and then back. Okay, you have a negative feedback to shut off the original pathogen. And there's people that years back had spent their careers just looking at this and modeling this, right, or doing biology on this. Then eventually we began to appreciate that inflammatory responses can cause damage to tissues, and the tissues produce molecules that are called damage-associated molecular pattern molecules that feed back on inflammatory cells to stimulate another round of inflammation. Okay, now this, and we'll talk about this in a minute, we can think of it as something happening secondarily, or we can actually think of the whole thing as kind of happening almost in real time. But functionally, this already adds yet more nonlinearity, right? Because now you have this big positive feedback loop, which I think is very key. Sort of this inflammation to damage to inflammation loop. Okay, so this already accounts for the bystander effects on your own tissues and so forth. But we also have to think about how does this get resolved. Well, it gets resolved through the introduction of anti-inflammatory mechanisms, sort of analogous to the pro-inflammatory mechanisms. They're really driven fairly early by the pathogen itself, but they're also secondarily driven by the pro-inflammatory mediators and mechanisms help to drive anti-inflammatory mechanisms. And of course, their job is to shut inflammation down, yet another negative feedback loop. And then this loop gets closed when we realize that the anti-inflammatory mediators are generally also the ones that the body uses to heal from injury. So this is how you can have a closed loop, and also makes it really, really hard to intervene and find only a positive outcome, because you need all the pieces to get from beginning to end. And moreover, I defy anybody in this room to trace through these arrows and tell me that you know exactly what the outcome is going to be, right? And this is about as simple of a cartoon as you can. It does a lot of interesting and powerful things. For example, it allows you to connect the original insult to the health outcome of the patient, which you could use this as a surrogate for, this damage or dysfunction, which is what doctors actually care about rather than the particular molecule you're measuring. They care about how you're doing. This allows you to calibrate models, for example, on data related to the pathogen and inflammatory mediators and predict the health status of the individual. And that is really the underlying basis of things like simulating clinical trials, which I'll talk about in a little bit, and work that we've done with Immunetrics. All right, so this is, let's call it sepsis, right? So this, as I've shown, is sepsis because you notice these arrows are not in balance, and so you end up in a state of persistent damage or dysfunction. We've been saying this for probably a bit over 12 years, and now we call this persistent critical illness. But this is really how it transpires, I believe, in the most simplistic fashion, not naming specific mechanisms or names of cells. All right, so what happens in trauma? In trauma, you have a traumatic injury, which pretty much directly damages your tissues, right? That's pretty straightforward. What does this do? Well, we now know that this also leads to a release of DAMPs because these are damaged cells. They're releasing these DAMPs that stimulate nearby inflammatory cells and also inflammatory cells that migrate into these injured tissues, setting in motion this positive feedback loop. Now, at the same time, trauma, hemorrhage cause a catecholamine burst and various other stress responses that drive IL-10, Th2 type responses, and you're right away having a so-called anti-inflammatory phenotype as well. And remember that anti-inflammatory also means you're suppressing the inflammatory response that you need to fight off infection. So you're kind of opening the door already to a secondary infection here, as I said before, nosocomial infection following traumatic injury. Now, these things are still happening, so pro-inflammatory mediators still drive anti-inflammatory. There's the feedback as well. And then, of course, we're still having to try to heal our tissues. We don't do such a good job, so we end up in this persistent damage or dysfunction. Now, one just very, I could almost call it trivial prediction of these pictures that I've shown you is that DAMPs in sepsis will be a late mediator because of the structure that I showed you. It'll come after that initial inflammatory wave, whereas DAMPs in trauma are predicted just by this picture to happen early. Same molecule in sepsis would happen as was originally shown by Kevin Tracy and his colleagues at around 16, 18, 20 hours, will show up in trauma at 2 hours. It confused everyone in the field as like, how could it be? Because they were naming these things late mediators. They're not late mediators, they're DAMPs, and they're produced depending on the exact set of circumstances at different times. So anybody who does modeling understands this inherently, but to clinicians and biologists, this was a huge conundrum, right? Now, there are many, many other subtle aspects to this picture that we typically use actually as a midterm exam on this course that is called the Systems Approach to Inflammation that I direct at Pitt. So we could spend a whole hour just talking about it. So what do we do with this? So we made 10 years ago, and this is, or actually now 11 years ago, we're in 2015, we made a very simple model wherein, as I said, it was a sepsis model wherein HMGB1, which is a prototypical DAMP, was a late mediator. 3 equations to the modeling people. You'll know this as a predator-prey model. Ridiculously simple. Physicists loved it because they like 3-equation models. This thing predicted a ton of qualitative features in sepsis. Everything from self-resolving inflammation, so each new episode of infection would be stimulating an inflammatory response that would clear the infection, come back to 0. You could overdo this and end up clearing your pathogen at the cost of killing yourself. You could have a situation where the infection overburdens you, and so you have persistent infection. You could have a chronic low-grade infection, or you could have a situation where your immune system is just insufficient, while at the same time having both pro and anti-inflammatory responses, which was also a big conundrum to the people in the sepsis field, because when they actually measured mediators in the blood of these patients, they would find often a huge elevation of both pro and anti-inflammatory mediators, and they said, how can that be since one is pro and the other one is anti, and anti should block pro, so those things shouldn't be there together. Well, when you do modeling, you understand that this, in fact, is completely related to the specific parameter values of an individual or a particular situation, and of course, in a population of patients, you're getting a range of responses. And so, again, a 3-equation model predicted pretty well qualitatively that you would get all these features, and importantly, makes it obvious that you couldn't possibly come up with a single drug that would treat all these people because they're all having morbidity and mortality for completely different reasons. The FDA still hasn't come around to that particular concept, even though people in the shock community have been trying to get them to understand this intuitively for, I'm guessing, somewhere in the vicinity of 20 years. Again, a model based on the simplest possible view of inflammation. Now, that's all good. It's nice. We could quit there, collect the Nobel Prize, and just sort of, you know, sit by the swimming pool. But we thought that that was a bit premature. So the idea is instead to go from there to making more quantitative predictions. And in this slide that I borrowed a while back, and I keep reusing, that I borrowed from Steve Chang of Immunetrics, or maybe John may have been the one that made it. So the idea is, how do you actually get this done? So for the non-modelers in the audience, when you're thinking about mechanistic modeling, the very first thing you have to go back is to the idea of objectivity. You have to objectively research the mechanisms that matter, right? And that may require a lot of time. This is the part that's actually the hardest of all. It's taking that and converting it into these sort of, well, some graphical view potentially, if that's useful, That's what we did. But some way of encompassing the knowledge in the most objective and yet somewhat abstracted fashion. Don't try to put every single thing there is, because not every single thing is a primary big knob that has equal importance to the other primary big knob. And try to think about compensatory things, like I showed you in those cartoons, is not having everything stacked to the pro-inflammatory side and nothing on the anti-inflammatory side, because for 20 years you've studied this one molecule and it's pro-inflammatory and you care only about that one, so that's the one you're going to put in your model, because all you're doing is perpetuating a situation that is really no different from you just making the hypothesis and doing the experiment without the model. You really want to think objectively about how are the different things canceling each other out. What could be there compensating? All right, so you go from this and you can develop representative models. We used equations, differential equations. Gary Ahn was a pioneer of using agent-based models, which can give you these pictorial type visual models. We've done a lot of agent-based modeling since then. It is a tool that can be very intuitive for non-modelers to work with initially, but also has many pitfalls and difficulties. It's very hard to do the next step, which I'm going to talk about, which is when you're collecting data, calibrating the models to the data. Calibrating agent-based models to data is very hard, and there's no automated tool set for doing that as of yet, whereas there are a lot of tools to do that for equation-based models. All right, so then when you do that, you can get things like this, where you have data and then you have some lines that are model predictions of a compendium of equations that describe the various variables in your model that now fit to the data reasonably well. And realize again that any statistical model will probably do better than any mechanistic model in fitting to the data. But your goal isn't to fit to the data perfectly. Your goal is to gain insights into the underlying biology that plausibly leads to the way the data look, right? And you're not really going to get that from a purely statistical model, but you will get that from a mechanistic model, and I'll talk about that in a minute. All right, so once you have that, you have to do something with it. And in our case, from a translational point of view, we want to make predictions in things like clinical trial simulations, because that's basically a prediction at the level of a population. Then, of course, you got to go back, collect the data, see whether you did reasonably well relative to what you predicted. And if you did, you're great. If not, you spend a lot of time recalibrating the model until you're doing better. And when you really can't recalibrate anymore, it means you probably are failing because you're missing mechanism. So you've got to go back and figure out what you're missing. But the good thing about this is it tells you roughly where you're missing and what you might be missing, as opposed to, I'm wrong. Well, you could be wrong for a lot of reasons, right? And so then either from in the typical reductionist biologist point of view, you have to go back and read every paper there is about everything and try to see if you can get a better idea to make your new hypothesis. In modeling, you fail, but you kind of fail in a limited sandbox that tells you you failed in a very specific area. Go look here and fix it. And that, I think, is very, very important. So this is an example of a study that, again, is now, 10 years old. Being able to do this, real data in genetically identical mice. And you notice that even genetically identical mice, given a complicated paradigm of injury, like in this case, a surgical cannulation and hemorrhagic shock, you get a lot of variability in the outcomes. And you also get it when you give them bacterial endotoxin in a quantitative dose. There's a lot of animal-to-animal variability that you almost cannot constrain as much as you may try. And there's some very interesting implications of that we can talk about. The bottom line is you can sort of do a reasonable job of fitting to the data, but fitting is not prediction. Fitting just says you're plausibly in the right space. Okay, so this is the time to start thinking about how it is that we start to get ideas for the workshop that we're in right now. So in the year 2003, a very, very accomplished graduate student by the name of Judy Day was working on expanding this mathematical model, actually with John Bartels. And this was in 2003. We were thinking we needed to include T-cell mediated immunity. And this was kind of because I personally had this idea that we wanted to go well beyond sepsis and trauma to use them as jumping off points for understanding acute inflammation broadly in other diseases and maybe going into chronic inflammation. And there were beginnings and hints. People like Jim Lederer, who's here, were already starting to talk about T-cells and their potential role. But this was very, very new, and nobody really thought this was a relevant thing until about 2 or 3 days following, let's say, injury or infection. So we had a very simple model with Th1, Th2 differentiation. Again, we didn't know about many of the lymphoid types that we now know. But we had that in there. And one interesting thing was to try as much as she did, Judy, it clearly disappointed me because I kept telling her, Judy, in endotoxemia, you get this Th1 followed by Th2 wave, and everything that everybody knows about trauma says that you should get something similar and that it should happen 3 days later. And instead, we kept getting stuff like this that kept saying that in trauma hemorrhage, there should be an almost very early and nearly simultaneous wave of T-cell mediated activity. And I said, Judy, you must be wrong, because that's actually always the right answer. The student is always wrong. And then we literally gave up. And there were no literature data. For us to be able to publish this paper, we would have had to get the entire shock field to completely upend its paradigm to be able to believe that this is true. The easier thing to tell us is our model is wrong, right? That's the easiest answer a reviewer can tell you, is you simply are wrong, because you can do anything with mathematics. Everybody knows biology is hard and fast, kind of like history. So we'll leave that alone for a second and come back to it in a couple of minutes. So in the meantime, we did what we could with the models, right? We didn't pursue that direction very much, but we said, what can we do? And with the help of Immunetrics and a lot of good colleagues at Pitt and other places, we pushed forward this idea of in silico clinical trials. The idea was to generate models that would be slight variants of a single individual model. They're deterministic, meaning we're not adding noise terms, but they're slightly different for each iteration because of parameter values, and that typically we equate with differential genetics. So we say you're slightly different in your response and your ability to produce a particular immune mediator, inflammatory mediator, say plus minus 10%. Well, we know genetics does that, in fact. And so we generated an entire simulated population. And that was the first paper of its kind. So last year we had our 10th anniversary of that paper in critical care medicine. More recently, in 2012, we made with Gary Neiman's group at SUNY and the Immunetrics folks, we made a 2-compartment mathematical model. So our prior mathematical models in mice were basically assuming the animal is one big bag and stuff floats around. So again, the people that do modeling, this is typical. But now you want to be starting to think more realistically. We have multiple organs that fail, and so we want to be able to start to have a way of getting a handle on that, not just the animal as a big bag. And we did this in swine, and we made animal-specific models. And remember that swine are not genetically identical the way mice are, so they're about as different from each other as people are, so this was actually a fairly tough exercise. We took that and we augmented this recently in a paper that's currently under review into 3 compartments, so that we have a tissue compartment where injury or infection can occur, lung where we measure various things but also as a surrogate for multiple organs, and then the blood, which is the communication medium across these components. And we made use of an ongoing modeling-driven 500-patient clinical study that we've done in trauma to try to see if we could create a virtual trauma patient from this type of model. And Tim will talk about these data in more detail. Suffice it to say that we have about a 4% mortality. We have predominantly males. Injury severity, which is measured by this injury severity score, is in the moderate to severe range. And then all of these folks, as I said, stay in the intensive care unit for 1 to 2 weeks on average, although they can go up to 30 days. And they also spend time, for example, using or making use of or requiring a mechanical ventilator. We'll come back to this survivors, non-survivors comparison in a minute. So one great thing about the way this cohort was designed, because it was designed from the ground up to support modeling, was the ability to create highly matched subcohorts. And what we did here is examined a subcohort of mild, moderate, or severely injured patients. And as you can see, the injury severity rises essentially linearly, age is the same, gender distribution is the same, various other comorbidities are the same. But as you can see here, their outcomes essentially vary as a function of injury severity, which is what you'd expect, right? But the good thing is this is your starting material for data, right? About 50 patients per group. And what that allows us to do is to compare model predictions to real data. So in this case, we have 10,000 in silico patients that were made from 33 models of patient-specific models that we generated, so from 33 survivors. And then using a parameter distribution, we can create 10,000 in silico patients. This is their inflammatory response in this case is started by the degree of injury. So here's their low, medium, and high severity. And this predicts that you would get longer, essentially, length of stay in the hospital, longer, higher morbidity as a function of injury. You notice it's not linear. It's not a linear connection between the injury severity and the outcome. This is what that table looks like pictorially. This is just to show that their ages were essentially the same, their injury severities as input, and then the outcomes. And notice that, again, the rise is proportional but not necessarily linear. This is nice. You could get 10,000 models to do what 150 patients do, but that doesn't automatically mean you're right, because this is a big and complex model. So here's another interesting feature. We built the model purely on data from survivors. Okay? The model predicted that 3.5% of these 10,000 people were going to be non-survivors. So just as a pause, I think it's essentially impossible to get a purely data-driven statistical model to do this. If it's never seen non-survivors, it will not predict non-survivors. So that already starts to highlight something mechanistic models can do. And the important part is that if you look at the current face of trauma, from what's published, it's 4.5%. In our own 500 patients, 4.4%. So that's kind of interesting. And so how did we get there? Well, the way you can do it is one of the ways you can achieve that in silico is to tune up parameters related to this key inflammatory mediator called interleukin-6. And you can convert a predicted survivor to a predicted non-survivor. So we said, let's look at that some more. So if we look at what happens in silico, you can segregate high versus low IL-6 producers. This would be their IL-6 levels, as you'd expect by definition. Low is lower than high. And you notice, though, that it's not everywhere and at all times, right? It's at the peak, but you'll notice a lot of overlap between the high and the low. And interestingly, you have a genetic predisposition to make high versus low IL-6 by a single nucleotide polymorphism, or SNP, and we had been measuring that. Again, it was part of the clinical study to start with. We wanted to be able to get SNPs in these key inflammatory mediators for the purpose of using them to segregate patient-specific models or group-specific models. A priori, 8 years back when we first started, so without even having all of this as an outcome or even knowing how it would turn out, we all rolled the dice and took a chance on structuring a clinical study for this purpose. Well, it paid off rather nicely because, again, you could segregate, in this case, 100 patients that we had SNP data on into low versus high producers. And here's some volunteers to give you a baseline, because no one typically takes a blood sample before running their car into a pole. And so that gives you the idea of there's roughly the starting point. Okay, again, fairly similar to what you see with the model. Now, this just obviously screams at you that says, hey, look, you can turn a survivor into a non-survivor by tuning up IL-6, and there's people with low IL-6 versus high IL-6, so obviously everybody with high IL-6 must have done worse, right? Because that's just obvious, because that's how life is. It's all linear. So instead, it's exactly not how it worked, right? So this is what happened in silico, which is David Brown at Immunetrics, when he ran all this, he emailed me saying, I think I did something wrong because I can't find any difference in the predicted length of stay of the high versus low IL-6 producers. And we had kept them actually blinded from the real data. And we sent them this, and it sort of exploded everyone's brains on the table, requiring substantial cleanup. So the bottom line is, in 10,000 simulated cases, you predict no effect. In 100 patients, when you segregate them by SNP, you find no effect. That's despite every other piece of this reductionist puzzle working exactly the way it's supposed to. When you put it together into a simulation that predicts the biology, you get the opposite of what you would expect. So we'll take a pause from that and talk about ways of doing modeling. So we've been doing mechanistic modeling predominantly, but data-driven models, which are association-based, are the typical way people do this kind of thing. They can be used in a more sophisticated way to suggest principal drivers and dynamic networks. They make, almost by definition, limited use of prior knowledge unless you're using a Bayesian structure and really adding a lot of literature knowledge into it. But the trade-off is you need much more data, you need a lot of data, and as I said before, you can only really predict within the data set that you have. You can't predict non-survivors from survivors if you haven't fed it anything on incidence of non-survivors. Whereas the mechanistic models, as I've shown you, they're highly causal. They can suggest all these non-intuitive things that I just have been discussing, like predicting mortality from survival or predicting that IL-6 won't matter a hill of beans when you just look across this entire population and segregate outcomes. And so they allow you to explain these types of emergent phenomena as we talk about them. And of course, this is much harder to do. This takes years worth of work to make one of these models. But the payoff is prediction outside of training data. And so the question is, can we learn more about trauma from data plus data-driven modeling? So this is a study that Andrew Aboud, who's here, and Rami Namas, who's here, and Ruben Zamora, who's here from Pitt, helped to put together, looking at that survival versus non-survival population. As I just mentioned, it's about 4% of the patients. But we can create a matched survivor cohort. As you can see, these are all of our non-survivors, and here's a matched set, so they're essentially identical with regards to gender distribution, injury severity, and if you see that there's differences in things like length of stay. Now, when you look at all this, you say, oh, it's obvious. The non-survivors stay less time in the hospital because they die, right? That's the obvious reason. And I'll show you that's not as straightforward an argument to make as you might think. For example, we put together a Kaplan-Meier analysis to look at just rate of drop-off from the study. And as you can see, the survivors and the non-survivors dropped off at about the same rate. And if you look at around day 7, which is where we have a lot of our granular data, their N numbers are really not that different. It's 13 versus 16. Importantly, they have essentially, because we structured it this way, the same degree of injury when you look across body regions. They're essentially identical in their injury responses. They're identical in their age, and they're identical in their genders, and yet one group of people lived and one group of people died. That's about as striking as you can get with regards to what the diversity of responses in acute inflammation can be. So we made use of the fact that we've been developing dynamic network models of inflammation for the last few years, starting from 2011 in mice, and then a bunch of papers more recently in humans. Key among them, and Tim will discuss in more detail, is this one, the study of nosocomial infection in the bone trauma patients that was recently published. And our goal is to try to go from patterns to networks, to use networks as potential biomarkers. And what I'll talk about in a minute is the idea that we can gain some insights into mechanism from networks. So we measured a bunch of things, in this particular case 27 mediators by Luminex, standard multiple inflammatory mediators that actually cover key aspects of innate and T-cell mediated responses, DAMPs like HMGB1, effector products like nitric oxide. And then we carried out this tool, this analysis that we published in 2011. We've made it available as a MATLAB code that allows pretty much anybody to take their data and get this kind of answer back. Not this beautiful picture. You get back a set of networks that you then have to manually create into these good-looking things. That takes time. But you can get network data quickly. And we can break it up by time. So in the first 24 hours, we find in the survivors this type of response, not much going on. You can think of it this way. And remember, these are severely injured people. And then a little bit more, and then a little bit more. And I think it's that when you look at all this, you see, first of all, this is the degree of connectivity of the nodes. So they're not super highly connected. These are not massive networks. And then the other part is that the word T-cell is all over this picture, right? So from as early as we can measure, you're seeing type 1, type 2 immunity, IL-17, so Th17 responses, and then these epithelial sort of stress cytokines that, again, I think Tim will talk about in more detail. So what does it look like in the non-survivors? Well, right from the get-go, their networks are bigger, and they get bigger, and then they got really, really big, and so we just put them in this kind of stuff happens kind of picture. And importantly, T-cell stuff is going on, but it's not as pervasive, actually, although IL-17 is very important, it looks like. And then later on only do you get this small subnetwork of IL-21, 22, and 23. And that's really interesting because this really great paper that was published from the Trauma Glue Grant Consortium, which is large-scale, multi-center consortium that involved Pitt. Tim was one of the collaborators. Jim was one of the collaborators. This was one of their conclusions. It said, and if you look at this, in severe blunt trauma, the early leukocyte genomic response is consistent with simultaneously increased expression of genes involved in systemic inflammation, which is what I just showed you, and in compensatory anti-inflammatory responses, so both pro and anti, which I said before, and we can see in the dynamic network pictures, as well as suppression of genes involved in adaptive immunity. So when you look at the comparison of the survivors to the non-survivors, you really look, it looks like actually a reduction in the overall degree of T-cell involvement. But is that really a bland response? No, these networks are quite big. In fact, we can put a metric of network connectivity. And you can see that the non-survivors have ever-rising network connectivity, suggesting a positive feedback, self-sustaining response. And how interesting is it that if you look at this and compare it to an index of organ dysfunction, this is the Marshall score, which covers essentially dysfunction across multiple organs, you see this rising multiple organ dysfunction that looks a lot like what was happening in the DINA curve when you compare the non-survivors to the survivors. And again, down below here, just to show you that our N numbers are really pretty comparable. So it's not that you simply have nobody left by 7 days, or that the groups are highly mismatched.
Hey, I think I have a good question that popped up that goes well with what you're talking about right now.
Okay, this current thing, or should I go back one?
The non-survivors versus survivors?
Yes.
Yeah. So the question is, did modeling of survivors versus non-survivors account for pre-injury general health variables like obesity, cardiovascular fitness, diabetes, et cetera? Yeah.
So we account for comorbidities in our highly stringently matched groups, and I think Tim's going to talk a little bit about that some more. But it's and a good example of it is in our Annals of Surgery paper that we recently published, as well as another paper in critical care medicine that came out last year.
And we'll try to get some of these papers up on the files. Some of them are already being posted. Thank you.
Okay. So, and I'll just sort of conclude with this. So an interesting insight that's been coming out over the last few years in the Th17 field, and this was suggested by Dr. Kuchru at Harvard, that there is this that not all inflammatory cells are created equal, and not all Th17 cells are created equal, and that you have this idea of pathogenic Th17 cells that are characterized by the production of GM-CSF along with Th17, and versus the non-pathogenic cells, which are Th17 and IL-10 producing. And so we said, can we just look at what we have? Could we infer that, these populations in our patients? We didn't actually go and do the flow cytometry to look at them, because we honestly don't even know where we should be looking. I mean, where are the cells that we should look at? Are they in the blood? Are they in the tissues? But we can look at the inflammatory mediators in the blood, and very interestingly, we find that in the non-survivors, the strong correlation of IL-17 and GM-CSF, and in the exact same non-survivors, you have a very strong negative correlation of IL-17 and IL-10, suggesting the possibility that from the beginning, what differentiates survivors from non-survivors is the presence of something like Th17 pathogenic cells. Something to think about. Correlation is not causality, but this is just the start. But the point is, hopefully, I've given you a sense for why we're excited about the idea that looking at lymphoid cells in acute inflammation and modeling what that might be doing is important. It's a highly underserved field. I think you're looking at the totality of the people that have an interest in that sitting around you. So I'll conclude just by reiterating that inflammation is complex and dynamic and nonlinear, and that you can't simply go from reductionist insights to predicting the clinical outcome. That, as I just said, and that modeling is probably very necessary to decipher this complexity, and that from a biology point of view, this focus that has been going on for a very long time on strictly innate immunity in acute inflammation might be insufficient, and that lymphoid cells might be quite important to be able to understand this both mechanistically and translationally. And I would argue that this could also go the other way, that the insights that we might learn about trauma and sepsis could be very useful to the people that are studying lymphoid pathways in chronic inflammation. I really believe that chronic inflammation is the chronic, constant restarting of acute inflammation, and that might be a very relevant concept to think about. So I'll stop there. I'll acknowledge a very large group of collaborators at multiple institutions, and thank multiple funding agencies that have helped support this work. And then for anybody who's interested, both Judy and I are members and co-founders of the Society for Complex Acute Illness. Our upcoming meeting is going to be in September in Cologne, Germany, jointly with the European Shock Society meeting. Eddie Neugebauer is the president, and Mark Megley is the organizer. They're great people that have put on this meeting jointly in 2008 previously. Tim and I wrote this review article in Critical Care Medicine that tries to concisely summarize some of the stuff I said. And then I'm very happy that there's enough interest in this that led to having one book in 2013 that Gary and I edited. Various people around wrote chapters for this. And then this book that just came out from Elsevier that tries to talk about the larger implications of modeling for the current biomedical enterprise. And I'll stop and take any questions, if there are any, or save them for later on. Thank you.